Claudia, thank you so much for joining us. Real pleasure to have you on, and very much looking forward to discuss all the work that you have been doing at Imperial recently. And firstly, I think on behalf of all the people who are suffering with illnesses like long COVID and CFS, I think we just want to say thank you as well for doing all of that work and committing to trying to find out more about this disease and hopefully pushing us in the way towards treatments as well. So just wanted to start off with thank you.
No, thanks very much. Oh yeah, it's a lot of work, but no, it's great to have such an involved patient group that are really sort of pushing us forward to get the best research we can.
For sure. So I thought we could start off with a little bit about your background and also, start us off with some of the research and work that you've been doing.
Yeah. So I have a PhD in viral immunology. I got my PhD at Trinity College Dublin, and then I moved to do a postdoctoral research role at Imperial 2 years ago. And since I've been at Imperial, this has been my project, or I've been on this project with colleagues. So, we've been using a cohort of patients from the FOSP consortium who were all hospitalised with acute COVID. So they had quite severe acute disease. Right at the beginning of the pandemic, so April 2020 through to September 2021. And from that cohort of people, when they got discharged, FOSP recruited them into this study, which means we've got really rich data on who they are, how long they were in hospital for, what drugs they were given. We know all about them. And then we followed them up 6 months after they were discharged to see how they were getting on. So there were about 2,000 people in that follow-up visit, that 3 months. And at that time point, we asked them to fill in 34 different questionnaires to see how their health was. And we took a range of different biological samples, so like blood and nose swabs and tissue samples, so that we could measure what was going on in their bodies alongside asking them how they were feeling. So this paper is looking at that first tranche of data collection. And what I first did with this data is I wanted to understand what is long COVID, which is a really difficult question. So within our group, the first thing we did was we used the most basic question in the world, which is, do you feel fully recovered? And anyone that said, yep, fully recovered, brilliant, you're my recovered group. And there are kind of standard then within the study for what should someone look like if they're recovered after hospitalization with COVID-19. Because when you go into hospital with an acute disease and it's quite bad and maybe you've been in the ICU, it's normal for you not to feel fully on your feet 6 months later. But what is not normal is to have additional symptoms outside that. So what having our recovered group does, it is allows us to say not just that these people aren't healthy, but they're not looking the same as those that feel recovered at that time point. It's not just comparing to a healthy population, because if you've been hospitalized, you might not look like a healthy population. And then everyone else that said they didn't feel fully recovered, we dove into those health questionnaires to see what sort of symptoms they were presenting with. We worked with clinical colleagues that run long COVID clinics and patient groups to see what common symptoms were coming up in people with long COVID. And there were kind of 5 main groups that we identified from those conversations. And so I looked through all the questionnaires and sorted people into piles. So those really common symptoms, and this is not to say this is not an exhaustive list, but were cardiopulmonary, so issues with lungs, hearts, feeling out of breath, coughing, things like that. Gastrointestinal, so any problems with stomach, like nausea, constipation, anything like that. Fatigue, so common. Anxiety and depression, so how it's affecting people's moods. And then cognitive dysfunction, or brain fog, so feeling like your concentration's not there anymore.
And is there crossover then? So obviously you have these different phenotypes, and then I'm guessing you're mapping the kind of crossover between different ones and how they... Those are the main kind of tranches that you found. Yeah,
they were the ones that we kind of identified. And then, absolutely. For someone to only present with one of them is super uncommon. So you have huge... imagine a really messy Venn diagram of everyone's kind of reporting slightly different things. But
chaos.
Absolute chaos. But I mean, that's the nature of it. So we are calling it long COVID, but these are not necessarily clinician identified long COVID cases, but it's the best we can do with the data we have. We don't have the resource to go through and interview each person to see if they have what their GP might call long COVID, but to be honest with you, if someone comes up to you and says, I feel fully recovered, you're not going to think they have long COVID anyway.
No.
And everyone else, we can kind of start looking for patterns of what might be elevated in those groups versus our recovered control group.
And in terms of the timings then here, because obviously you're saying they were discharged from hospital, right? Obviously some of them you catch up with them later on. Everything seems fine. And then there's a subset, obviously, who are still having ongoing symptoms that aren't the normal recovering from hospital.
Yeah.
But because obviously this is something that we were discussing before that it's very difficult to measure and is often can be quite misleading, right? Because I think on the kind of NHS technical definition is the kind of 12 weeks symptoms 12 weeks or more post-COVID constitutes as long COVID.
Yeah.
And obviously I think that can be a little bit misleading because you can get post-viral fatigue, which lasts 12 weeks to 6 months or whatever. I had that following glandular fever, for example. But there's something kind of fundamentally different, isn't there? That feeling of post-viral fatigue versus a kind of more chronic type of illness. And then you obviously go into the descriptions of chronic fatigue, which is then kind of post 6 months of having these types of symptoms. So where does this fit within that framework? And at what timings following hospitalization were you asking them that question?
So our cohort, in the paperwork, it says 3 months first research visit. That was supposed to be the earliest time people showed up for their follow-up to take blood samples and give questionnaires. When we did the analysis, we restricted the analysis to only people that had come to us 3 months or longer after their hospital discharge. We did have a few people that showed up like a month after they were discharged, and it was like, that's a little bit too soon to really be able to quantify this. On average, our people came back 6 months after discharge, but the latest was 8 months. So we have a range from 4 to 8 months, with the average being 6 months post-hospitalization. Yeah, the definition is an exceptionally thorny issue. So the WHO, which is the World Health Organization's definition, is any persistent symptoms 3 months after your acute COVID infection. I think the CDC and the NHS is 12 weeks. So yeah, it's about the same. But the timings of it do vary country to country. And it's just any new symptom, which I don't love because there are so many things that can happen to a person like over time. As far as like chronic fatigue syndrome and the 6 month cutoff, personally, I view long COVID as a type of chronic fatigue syndrome. I don't think they're the same, but I think they're under the same umbrella. I think the difference with long COVID is we know what the causative agent was, and generally we know when that happened. Whereas chronic fatigue, it can be anything, and because it's a process of elimination to get a diagnosis, you only get that diagnosis quite late on, and we have no idea what the initial trigger incident was for each individual. So long COVID research is difficult in a way, but it's very clean.
Yes.
And I mean this in the nicest way possible, but we have an entire population got infected with the virus for the first time. So this isn't reinfections because COVID-19 didn't exist, SARS-CoV-2 didn't exist. With our group, we know when they got it, so we know what strain they got. We know how long they were in hospital. We know the exact dates. So what I hope is we're looking at early in the development of chronic fatigue syndrome type thing. And that might be able to give us a bit more insight into like what's happening. Because with chronic fatigue syndrome, because you only get that diagnosis at 6 months, and some people it can take way longer from their symptoms to get that diagnosis, the causative incident is so far away that your biology, like your immune, if it is an immune response thing, and I'm biased because I like immunology.
That's your money, your routing.
Like things might have changed so much that it's going to be much harder to untangle the web because there's been so much time passed. Whereas long COVID, we know what's happened. We've got a really clear timeline. We know people have not seen this infection before, so you're not seeing like layers of seasonal infection on top. So it's a lot cleaner of a picture.
And we are essentially the perfect guinea pigs now. But I think that's actually it's very good to hear, and actually quite nice to hear that kind of distinction, because I actually see a little bit of tension, and often there's a little bit of a rift between whether people are talking about long COVID or chronic fatigue syndrome. And either group saying, don't leave us behind
Yeah.
or don't leave us behind. And I think that is quite important. And yeah, the way I kind of see it as well is we know the trigger, right? So chronic fatigue can be from a range of things. I think, bacterial, other viruses, stress, a whole variety of different things. But with long COVID, like you said, we have a specific trigger that we are aware of, aren't we?
Yeah. It just makes it a lot nicer to work with from a timelines perspective, because often if you run like any sort of experiment, your timings are so important. You're like, I have my plate of cells and I'm going to treat them with something. I'm going to come back 6 hours later. With long COVID, we know when the insult happened and we can see what happens sort of 6 months down the line. It's just a lot easier to manage like how you're understanding that data, whereas chronic fatigue syndrome has been a very difficult thing to do. And people have been working on chronic fatigue for years. Like concerted effort has been put into chronic fatigue. I hope that the research we're doing on long COVID supplements, supports, and drives forward the chronic fatigue research as well. Yeah, and I know that when I first started this position, the first thing I was doing was going through and reading as much chronic fatigue research as I could to get an idea of what we knew about that. And at that stage, I wasn't completely sold on the idea that they were the same thing. And I still don't. I think there is clear water between them, but I suspect there's kind of shared pathways involved. Because you want to know what's already been done. You don't want to reinvent the wheel. You want to check that you're kind of adding to the knowledge base and you understand what you're doing. And I know that some of the long COVID work now as well, they are trying to include chronic fatigue syndrome cohorts as well.
Yeah.
Partly as like just a comparator marker. But I think that'll be really helpful in understanding what's driving both syndromes.
For sure. And I think, like you said, because you have this known causation and you have this known date of infection, then you can really look into the mechanisms and see what hopefully is going on. So speaking of mechanisms, let's go into some of the results of the research then that you found. Walk us through a bit of the study and what was discovered. Yeah,
so we had our 5 kind of, I'm going to call them endotypes, phenotypes, subtypes of long COVID. And in each case, we ran a huge panel of protein markers in the plasma samples we got from participants. So we ran 368 different proteins to see how things were changing between those different groups. And then using what I'm going to call fancy maths. So it was penalized logistics regression that took every mediator we had from each of our participants and then compared it against the recovered group to see which proteins were higher respectively across those two groups, and then how they linked together. So, for example, one of our most commonly upregulated proteins was called matrillin-2. And so doing this penalized regression showed that matrillin-2 was pretty much always higher in our symptomatic group compared to our recovered control. And the penalized regression basically meant that we were sure that this was a real thing and not just an artifact of comparing to, if you compare things over and over again, eventually you're going to see something. So doing penalized regressions allows you to be kind of more confident that what you're seeing is real rather than an artifact of like how you're analyzing the data.
Okay. And was this for a specific phenotype? Sorry, was it like the GI group
each of them? So I did each of them in turn.
Okay.
So in each case, comparing to the recovered. But as you said earlier, we don't get one symptom for long COVID. Everyone's in the different groups. So we did have a lot of overlap between groups, particularly the cardiopulmonary and the fatigue group had, I can't remember the percentage, but in the paper, I've got a graph that shows like how the groups interact. But a good number of our cardiorespiratory people also showed up in our fatigue group. Okay. So that makes it harder to see specifically what's driving one group versus another. But it does allow us to see what's driving symptoms versus recovered and gives us some idea, because there'll be some people that have cardiopulmonary and not fatigue. And so then any differences between the cardiopulmonary and the fatigue group versus the recovered, are being driven by this very small group of people that have one symptom and not the other.
Yes.
Does that make sense?
Yeah.
So doing that in turn with each of our symptomatic groups, we got a bit of a picture of what might be going on. So the most common proteins that came up as being upregulated in the symptomatic group were all involved in a cell type called myeloid cells.
Okay.
So these are a type of immune cell. They're a precursor to a few different immune cells, including monocytes and dendritic cells. The important thing to note is these are a white blood cell. They're released from your bone marrow, the myeloid cells, and then when they hit your tissues, they start differentiating into these more specialist types. And you get an upregulation of the release of these cells when you've got an infection. Like they're notable that you get more white blood cells when you have an infection. What's interesting is that these myeloid cells, or like these markers that are suggestive of myeloid cell activation, was only upregulated in those with symptoms, not just those that recovered. So this is why it's so important that we have our recover group that are in hospital at the same time. Like there's no reason why these people should look different except for the symptomatic differences. So that suggests that maybe there's something going on with myeloid cells that means they're overactive in people with symptoms.
Okay.
Next question is, why are they more active? We don't know. That's the kind of next thing that we want to do. But this sort of research is great for like exploratory work. So it could be that you've got persistent virus.
Mm-hmm.
And so that's what's triggering your white blood cells to be more active and to do things and release these proteins that we're picking up in blood samples. Could be that there's some sort of like autoimmune stuff going on that is activating these cells. It could be epigenetic changes at the DNA level that's impacting how regularly these cells are released from the bone marrow.
Mm-hmm.
Like we don't know. And all these things are being kind of looked into by various different groups globally. Like it's all kind of hot stuff. It's all happening.
Okay, so just to clarify, so with the myeloid cells, they are a precursor to other white blood cells like monocytes, etc. And it is normal to have these myeloid cells.
Oh, absolutely.
Like when there's an infection, for example, you've got a virus or the flu.
Yeah, and you'll have them, they'll be released to kind of at a steady state all the time. Because your body wants to be prepared for any sort of infection or like assaults. But it's quite energy intensive to mount an immune response all the time. Yes.
Okay.
And that's one of the reasons why, like, if you have flu, I mean, there are quite a lot of reasons, but it's one of the reasons why if you have like a bad flu, you're really, really tired afterwards because your body has just spent a week mounting quite a significant immune response to try and clear everything out. So if your body is continually mounting an immune response, that is going to make you tired.
Yes. Yeah. Okay. That's a very familiar feeling to a lot of people watching, I think. So it's essentially a constant. So, and I guess this is also why there's this sort of notion towards whether it's viral persistence or autoimmunity, because there's this low grade release of these myeloid cells, which is essentially a low grade activation of the immune system. Is that right?
Yes. However, I should say that because we were just measuring soluble proteins in the blood rather than the cells themselves, I can't definitively say we have more myeloid cells being released. I can only say that there are proteins that are suggestive of myeloid cell activation. Which is like a nuanced difference, and it's something that we'll be exploring so we can say whether or not it's definitely we've got more myeloid cells.
Okay, and across the different phenotypes then, what does that, because I speak to different people and you have a whole range of different symptoms. Some people, like you said, are very like neurological, so they really struggle with brain fog, but then they'll be like, I don't actually have any fatigue, or I don't have any GI issues. Although I think across the board, like you said earlier, the fatigue one is I think it's probably 80-90%.
Yeah, it's definitely up there.
It's very consistent. So how does this data present across the different phenotypes? So from one to the other, and obviously it's difficult to specify like where there's a separation, but in general, is there like masses more for neurological or?
So the really interesting thing, so all of them had this background of the myeloid cell story.
Yes.
And then each group kind of had its unique thing. Less so cardiopulmonary and fatigue because they were so like one generally came with the other. But people with gastrointestinal issues had upregulation of a marker called secretogranin 3. Which is also known to be upregulated in people with IBS. So it makes sense, right? It's I'm not saying it's causative, but also a marker that is known to be associated with GI problems was also upregulated in our people with stomach issues. So it kind of tracks. People with cognitive dysfunction, brain fog, had upregulation of a couple of proteins that are involved with neural regeneration.
Okay.
So in their blood, we are picking up signs that neurons are trying to like regenerate. as they try and kind of like bring themselves back to homeostasis. So they were the kind of the main ones. Our anxiety and depression group were actually quite small. Yeah, so I can't really speak to that so much. That was our smallest group, if we saw any major changes there. But all of them, we saw the myeloid story. And then the other thing we saw was proteins are associated with the upregulation of complement.
Okay.
Now, complement is probably one of the oldest arms of the immune system. It's like even corals have a complement system. It's a really basic way for any organism to recognize something not self and respond to it. So it's really basic. Everything has it. And seeing changes in that kind of also suggests that like this very basic thing is being it's slightly perturbed in how it's working, which is quite interesting. And there are people, again, actively looking at complement, like specifically, aside from this.
So do those two, with this stimulation of the complement immune system, does that usually, in general, go hand in hand with the myeloid when you're ill, for example? Is the complement, so it's not?
Well, so when you're ill, you will have your complement pathway will kick in, and we will see upregulation of certain markers. And the thing that there are a couple of things that activate the complement pathway. One of them is antibodies, which fits in with, there's kind of been an autoantibody story with long COVID, that potentially things are being caused by the release of antibodies that target self rather than that are targeting the pathogen. And because complement can get activated by antibodies, that kind of fits in with that story. And then the complement can also be activated by pathogenic markers. So again, could be viral persistent. Maybe there's something going on that's activating that complement pathway, and then we're picking up the products of it in plasma samples from those with symptoms.
Okay, because there has been a few bits of research which have demonstrated there's been kind of residue left over in certain areas of the body. But because this is something actually I've had a question about, is when it comes to this viral persistence, I've also read that viral persistence is a relatively normal phenomena with other illnesses like glandular fever as well. So you would have remnants of the virus within you, but it depends on how your body is reacting to it. Is that correct or not?
So it's very virus specific. There are some viruses, like herpes viruses, which cause, like, among other things, like cold sores. And the cold sores, once you've got them forever, and they'll come back when you're stressed, and they'll come back at the worst times. My
mate's like that. Whenever he's skiing, he'll just break out with...
So that's a virus that is able to, like, get into your cells and, like, live there permanently. Chickenpox is the same. You can get reactivation of virus. It goes and lives in your nerves. And then you can get reactivation of the virus, and it will come out as shingles, and you'll get, like, really... the classic is bands around your torso as the virus, like, shoots down the nerve until it hits the skin, and that's when you see the lesions come out. So there are viruses like that are actively in you. And a human papilloma, another classic one. So we now have the vaccine against that, but that gets into your cells and can cause mutations that can lead to cancers. So there's those sort of viruses. And then there's things like SARS-CoV-2 and flu and RSV and loads of other ones, which don't do that. They won't get into the cell. They won't live. Well, they do get into the cell, but they don't intercalate the DNA in the way that those other viruses do. So you're right in some ways that you can get viruses that stick around for life. SARS-CoV-2 isn't one of those. So the theory is that potentially there's a viral reservoir somewhere within the body, like the virus is getting into a place, but it's continually replicating. It's not going dormant. It's, like, continually active and alive, like, in a space.
Because if it went dormant, that wouldn't be a problem. Is that right? It would be kind of benign.
Well, it would be the cold sore phenomenon. Of it would be fine until it woke up, and then you'd have problems again. So it wouldn't be good. But it's a different type of virus. Like, the way the genetics of this virus works, it can't do that. So once you clear it, it's cleared. It's gone. So if we're still picking up remnants of it, like, once you've had a viral infection with this sort of virus, you can pick up proteins of it for a while after it's been cleared. So if you've had COVID-19 and you come to me and I swab you a week later, I might pick up viral proteins in you, but I won't be able to grow virus from it. The virus will be dead. It's just that it's still kind of like debris hanging around. But with COVID, the idea of the viral persistence is that it's in a site in the body and it's still replicating and it's still releasing virus into your body. We actually, to check this, because a lot of our stories kind of pointed towards there being virus around, like it's the most obvious answer is you've still got virus there. We had swabs from people's noses when they came back to give us the blood sample, so they're taken at the same time, and we couldn't detect any SARS-CoV-2 antigens at all in those samples. That just shows that it's not. That doesn't necessarily rule it out, because it just shows that it's not where we swabbed for it.
Okay.
We didn't take. There's been some evidence that it can replicate in the gut as well.
Mm-hmm.
So we didn't take stool samples, for example, and potentially if we'd have taken stool samples, we could have seen something. This is the problem with you have to plan your experiment. You have to plan your experiment, and then you do it, and then with hindsight, you go, "God, I wish I'd taken that sample as well."
And then hindsight is a wonderful thing.
Then you set up a new study.
Yeah. Okay, so essentially, though, across all the phenotypes, across all the patients, essentially what we're seeing is this kind of marker that is suggestive of myeloid activation and essentially a kind of low-grade immune system that is firing. How does this tie into one of the most bizarre symptoms, which is post-exertion malaise? So when you have people who maybe they have low-grade symptoms, or maybe they have relatively little symptoms, but any kind of exertion, whether it be physical, whether it be mental, after that, they will have this almost flu-like reaction and come down with essentially an illness again.
Yeah, I'm going to be really annoying. I'm not a clinician, and so okay. I can't really answer that. I don't know from what I've done.
Yeah. Okay.
Sorry.
Okay. But it could be, I guess it would tie in with the fact then that it's maybe causing that stimulation even more as a result. I think that potentially,
and it could be, it could be nerve involvement as well. I like it. There are so many things that it could be, but it's not quite my area of expertise, so I wouldn't want to say too much.
No, that's all. That's all good. In terms of, I noticed that you looked into the differences between male and female, because I think that's something that's been brought up a lot and often gives us a little bit of clues as to why it does seem that it's skewed more towards females being affected. I can't remember the exact statistic, but it's something around maybe 70%.
Yeah, it's something like that. So in our study, because it was generally men actually that had most severe acute disease.
Okay.
So in our study, it was something like 65% of our cohort is male. But then when you look at those with long COVID, I think 45% are female, which sounds like, oh, it's 50-50. But like when you take into account the fact that our starting population was overwhelmingly male, and now we've got to almost parity with long COVID symptoms, like being female was such a risk factor for developing long COVID symptoms. It's a really good question, and we don't have any good answers currently. We did a little bit of stratification against sex, and we split it by age because we wondered if maybe it was a hormonal thing. So we took and I myself and my co-first author are both women, so I apologise to all women, but we just went 50 menopause. Right, we're going to look at people. We're going to look at men and women over 50 and look at some of these markers and see how they change, and men and women under 50 and see how they change. And there was no difference when you split it by age, which would say to me that it's not a hormonally driven response. Although I recognise that 50 is not a fantastic way to deem menopause, but we hadn't asked that question on the questionnaires, so we were doing our best with the data we had. So yeah, I don't think it's necessarily hormonal. However, we touched briefly on like autoimmunity, and that's one of the things people think might be driving long COVID. Women also have a much higher risk for autoimmune diseases.
Yeah.
So that kind of ties into that story, that maybe that is what's happening, and it is a form of autoimmune, or at least there is an autoimmune component to it, which would explain why women are so much more high risk than men to end up with long COVID. There's more work being done on this. And a lot more to do, because we don't really understand. Well, there are a few theories why women get more autoimmune diseases than men, but it's incompletely understood.
Yeah, it's still something that's in progress.
Yeah.
And in terms of, because one of the things that jumped out actually when I was reading the study, so obviously you mentioned the hospitalization, and that allowed you to track exactly when they'd had the virus, and then obviously their treatment and then their recovery times. But what we're seeing with long COVID is that a lot of people are not necessarily hospitalized, but we're seeing people who maybe had a very mild infection, and then later on they potentially had some kind of trigger, which then led them to come down with long COVID or other chronic illnesses. How do you sort of account for that within the study, and how does that translate to the kind of wider population?
Yeah, great question. So our cohort they were all hospitalized, which is great in some ways, but also the majority of the population was not hospitalized with COVID-19. Yes. That is not the norm. So we dealt with this in the paper by looking across the different severity groups, because there was a thought right at the beginning of all of this that long COVID was linked to how severe you were, and the more severe your illness was, the more likely you were to get long COVID. And so we compared the so everyone that comes into hospital with COVID-19 got a WHO, World Health Organization severity score, which just reflects the amount of support they needed. So that goes from just needing fluids to needing an oxygen mask to needing like nasal prong oxygen to being like on a full ventilator. And looking at those different WHO severities, there was actually no significant difference between any of those groups. So you're no more likely to end up with long COVID if you were very, very severe, or actually you probably could have managed your COVID at home, but maybe you had like a comorbidity that meant your doctor felt you needed to be in hospital with it. So that kind of tells us that severity has nothing to do with it, which is borne out in the general population. We are partnering with other studies. So there was a very large study called REACT. And when we ran the analysis for the study that we just published, we actually included all of their samples in it as well. So everything was run together. We mixed them all up. We ran them all together so we know, so that the samples are as comparable as possible. And one of my next projects is going to do an integration of what I've just shown with the hospitalized cohort and our community-acquired cases. So REACT was all from the community, and they took samples in the height of COVID. They had nominated households, I think, and they were going around to swab. And if you tested positive for COVID, regardless of if you had symptoms or not, they take a sample. And then from then, they've gone back and they followed up. I think it's 3. Actually, I'm not even going to say it. They went and followed up at 2 different time points. Yeah. And so we can then look at those and compare. So I think the hospitalization, I will hope it will map onto the community cases. But that work is in progress. Yeah. REACT are also doing their own independent analysis of their data. So yeah, keep an eye out for that, because that will be really interesting to see what this large community study looks like as well. It was huge, wasn't it? I remember seeing about it. They had a ton of data, a ton of different people, and that kind of brings me on actually as well quite nicely to the community itself, because we spoke about this again before, and that is one of the benefits in many ways around the patient community existing within long COVID or chronic fatigue, is that the patients are sadly very desperate, but in many ways they are incredibly engaged and willing to get involved in trials, share data, collect data, and contribute. Was that something then that you found within your study? And how can we engage people even more from a research perspective going forward? Yeah, so the community is amazing, and I think it's great. It's great for researchers because we have so many people that are interested in what we do and are also helping guide the research and making sure that we're looking at the things that are important that need solving, because there is a whole long laundry list of things that we want to get done. Yeah. But engaging with patients allows us to prioritize that, to make sure that we're making sure we hit milestones that people really want to know the data from. So within FOSP, we have it's public patient engagement. It's called PPI, and I cannot remember what that stands for right now. But it's, we work with a number of different charities that have an interest in long COVID, and also it's the Long COVID Support Network. And we have regular stakeholder meetings where that group is shown the data that we've done, but is also asked for feedback on areas that we can do. And they're also our first point of call when it comes to new studies. So if people want to get involved in new studies, I would really recommend, be part of a community, talk to each other. There's also a website. So if you search for Be Involved in Research UK,
Sure.
That is a nationally NHS run website that lists all the different trials that are happening, and it works for everything like cancer, heart disease, whatever you want, and you just put in what you're interested in being involved in and your city, and it will give you a list of the trials and studies ongoing if people want to be more involved in research. The community it's really good, and it also focuses our attention, and I'm really glad that Twitter can be a horrible place, but it's also really nice to have a community there where people can reach out to each other and talk to each other, because I think as much as we don't understand long COVID as much as we would like to, it is a lot worse to have a condition and not fully know what's going on and not know what the answer is going to be and not know what the outcomes are. Yeah. So I'm really glad that people have that community and can talk to each other. It definitely supports the research that we're doing. And I mean, the FOSP cohort that I was working with, we had 10,000 people in the first tier who, after coming out of hospital, agreed to talk to us, which we really appreciate. And then a further 3,000 people that were willing to get poked and prodded and fill out acres of questionnaires. And we absolutely could not do this without that interaction.
That is crazy. And I think that is a testament to the community, because they're so engaged and so willing to be a part of that. In terms of some trials then that you're excited about coming up, and then we can go to some of the outcomes as well, and in terms of potential kind of areas that we would want to look into as a result of your study. What are some potential clinical trials that are coming up that either you're involved in or you're very excited in terms of seeing the outcomes, seeing what it might reveal about mechanisms, about treatments, about potential kind of therapeutic and diagnostic pathways? Yeah.
So there are a couple of clinical trials looking at antivirals going on. I think these are all fully recruited and it's all in play, which is great because it means we'll get the data hopefully sooner rather than later. So looking at antivirals, because if it is just that people with, or at least some people with long COVID have got a viral reservoir, giving them antivirals for 6 months should clear the virus, and you should see an improvement in symptoms. So that will show whether or not that is the case in some people. So I'm really eager to see those results. There's also a clinical trial about to start it's a branch of the FOSP study. It's called FOSP-I, based in Leicester. And they're going to be using a anti-IL-6 treatment, which is basically a way of turning down the immune response. So it's a drug that is currently used in rheumatoid arthritis to stop the immune response from going so hard and causing more problems.
Just chill out.
Just calm down. That's starting. They're starting recruitment for that soon, and I'm very excited to see what the results of that will be, because again, that would give us a treatment that we already know is verified, it's well used, it's well tolerated in the community for a different disease. So you'd get that out to people relatively quickly if it comes off. That's a few years away. We're starting recruitment for that trial. Well, the people that I work with are starting recruitment for that trial shortly. So it will take time to do that properly, because you want to make sure you're not causing any additional problems and you're getting benefit from a treatment. And it's
But that repurposing is quite a good thing, because obviously drug development, for example, in its entirety is a decades long process. And if we can repurpose certain drugs that already have a tried and tested safety profile, we can test ones that make sense, and we can see very quickly, relatively, which ones are going to be useful, and then you can assess how it goes within the community. In terms of biomarkers and kind of diagnostics, I wanted to dive into this a little bit, because one of the major issues at the moment is obviously it's hard to quantify what do you have. It's a process of elimination, as you said beforehand. It's hard to quantify exactly what is wrong, whether you have long COVID or chronic fatigue, whatever, what sort of phenotype of that you have, and to what degree you have that. So having a biomarker where we could say, okay, there's this blood test, and from that we see that you've got this subset of long COVID, you need this treatment, this will work best for you, whatever that combination of therapies may be. How does your results tie into that and any future work?
So biomarker, I think, will be so important. It will just make everything a lot cleaner, a lot easier to understand. I suspect that for long COVID, we will never have like a single biomarker. I suspect we're going to have, I don't know, if we could get it down to like 5 things that we're measuring.
Yep.
And then you can say like, okay, your 5 things are all skewing far enough away from the norm to say that this is long COVID versus anything else. That would be ideal. We are actively looking into this. There is work being done on this. So the way that it would be done for this is we'd get blood samples from a load of people that have confirmed long COVID. I would hope a load of people that also have confirmed chronic fatigue as a group, and a load of people that have like various other diseases. Because the work we've done, I'm very proud of it. I think it really helps like move things along further. But a lot of the things we found are also commonly seen in other inflammatory diseases.
Okay.
So it's very difficult then to discriminate between someone with long COVID versus someone with like acute flu, for example.
Okay.
And you wouldn't necessarily want to treat those 2 people the same.
No.
So to have a good biomarker, it needs to be specific and it needs to be sensitive, which is why I think we're going to need multiple things that we measure at the same time to pull you away from other diseases that might have a similar profile. But yeah, the way that we'll fix that problem, because we need something, would be to recruit long COVID, chronic fatigue, and healthy individuals, and some with related but different diseases, and take blood samples, see how different proteins are skewing, and identify then a panel of markers that we can use as diagnostics. But I think that's a little way down the line. But it is, we've I don't know if I'm allowed to say this, we're in process of writing grant proposals to get that sort of research done. Okay. But it's gonna take a while. Yeah, I mean, chronic fatigue, again, we don't have a good diagnostic for chronic fatigue either. No. So it's tricky. It's tricky. And it's not that we don't have those things because no one's tried.
No.
It's because it's challenging.
It is very welcoming to hear, though, that, like you said, there's proposals in progress. There's already trials in progress. I think actually I did see a couple of those about the antivirals. The results are coming actually, I think maybe even in 2024 at some point, end of the year. So it is nice to see that there is quite a lot of work going into this. And then also with the biomarker, I think that will be an integral part of allowing us to, if we need to develop further treatments or develop drugs specifically for that. If you have a set biomarker and something that drug companies can agree on and say, okay, this is what we're targeting, and this is what we're going to measure as to being successful or not successful. A very similar thing happened with chronic kidney disease. I'm on a trial for that at the moment, and there was no progress for years and years and years. And then they reached the point where they actually found a biomarker, which was protein within the urine, and they agreed on that, and they said, this is something that we can agree on that we want to try and reduce, and this has a strong correlation with a positive long-term outcome. And as a result of that, there's been about 20 new clinical trials that have come out in the last 2-3 years. Incredible results. I mean, from my own one, I'll share a graph of it, but it's actually crazy how it's gone down. And it's very exciting to see that same tactics might well be applied to long COVID and chronic fatigue as well.
Oh, absolutely. And I think so that our research, the fact that we had all this myeloid cell story, even if that specifically does not give us a biomarker out of it, it at least tells us there's something going on with the myeloid cells. And then you can track back and you can try and understand what's happening, what point might that have happened, and you can then start looking, and is there anything in there that you'd want to target? Because the immune system is very well balanced. So if you push a lever, something else might go a bit haywire, which, as we said earlier, is why using drugs that already exist. Yeah. We've done the work. The work. If you have to start something completely from scratch, which drug companies will do, and I'm sure they'll be brilliant, but it's a very long process. Yeah. Because you need to make sure it works. You need to make sure it's safe. Mm-hmm. Then you need to make it affordable. Then you need to get it out to people. Yeah. And that's a process.
Yeah. So I think in the meantime, we can chuck some stuff and hopefully see what sticks.
Well, there's a lot of scope, I think, for work to improve it, and I know that there's a lot of people working on it globally.
Yeah, that's amazing. And a final one, because I saw in your research you talked about potential therapeutics as a result of your research and targets. And I noticed something about JAK plus inhibitors or something along those lines. I don't know if you could maybe give us a little bit.
Yeah, I can give you a little bit. So I think so the JAK-STAT pathway is one of the most like integral. I'm slightly biased because I did my PhD on, but the JAK-STAT pathway is like really important in your response to a viral infection. So when a cell notices that there's something non-self, there's a virus, there's something going on, it will release these proteins called interferons. These are a cytokine, and they act on neighboring cells to put them on high alert that something's happening. Okay. So you've got your one cell that detects pathogen, and then all its neighbors start like ringing the alarm bells. So the interferons hit next door. They start this JAK-STAT pathway, which is think of it like you know those like pegboards where you can drop a coin in and it hits a load of stuff on the way down. Yeah. So that's your pathway going down, and the JAKs is one of the pins in it. So that gets activated. If you can tone down the JAKs, you can stop the coin going any further, and you can stop the inflammation happening. So the interferon hits the top, starts going down. Ultimately, that results in gene expression, and you get the release of these things called interferon-sensitive genes, which they do a whole. There's over 500 of them. They do loads and loads of stuff to stop viruses from infecting the cell, which is a good thing. But as I said before, mounting an immune response is really energetically exhausting. Yeah. And that goes for individual cells as well. So if your cell is continually producing an immune response, it will get exhausted and it will die. So the interferon response is great for stopping viral infection going any further, but can have consequences for that tissue. So using a JAK inhibitor would dampen down the immune response to the pathogen, and I'm using pathogen quite loosely here because we don't really know what's going on in long COVID. Yeah. So if it's just inappropriate signaling, it would also turn off the inappropriate signaling that could reduce the immune response, that could then help with symptoms. But again, if you turn off your immune response, that causes other problems. So I don't know. There would need to be some fine tuning of it to work. You couldn't just say if I gave you a pill that turned off all your JAK-STAT signaling, you would encounter issues exceptionally fast. Yeah, okay. So it's something that needs to be fine-tuned, but it's an avenue that we can kind of potentially look at and try and get something that would sort of dial back that immune response. It would work in a similar-ish way to the anti-IL-6. Different pathway, different cytokine, but same idea. That drug also turns off a little section of the immune response to kind of help the body get back to its equilibrium where it wants to be.
And kind of dampen that cascade, really.
Yeah,
Amazing. Well, thank you for explaining such a complex topic so concisely. Claudia, thank you so much for your time. I really, really appreciate it. Very much appreciate all the great work that you're doing. Are there any links that I can leave in the description below?
Yes, absolutely. So the FOSP network, who is the main study that I've been working on, they've been fantastic. We've had great patient involvement. I'll give you the link as well for the clinical trial research if anybody wants to get involved in studies in their local area. Don't feel you have to, but if you'd like to be involved in research, we'd love to have you. Yeah, I can probably think of a few more, but yeah, those are the main ones.
All of those links will be in the description below, and yeah, I hope you guys enjoyed this episode. Any questions, just let us know in the comments, and I'll see you next time.